Did you know that strong social bonds can be as good for your health as quitting smoking? Or that connecting with someone even briefly in a checkout line can help to improve overall happiness? There are reward centers in our brain that get a little boost whenever we give to charities or we help someone in distress. Social connections hugely benefit our health, but there can also be costs when we lose these connections. Loneliness can increase a person's risk of death by 26% in any given year. We all know that humans are social creatures, but actually many of Earth's creatures lead social lives. Many insects, vertebrates, and even many microbes live and interact in social groups. And for each of these species, social interactions are tightly linked to their individual success. Their social partners can help or hurt as individuals attempt to find food, avoid predators, and secure mates. The thing is, we don't yet understand the mechanisms that underlie social behavior. And this means that when these mechanisms start to go awry, or even when we struggle with social interactions, we don't necessarily know how to help. But what if we could understand the mechanisms that underlie social behavior? Imagine knowing that core set of molecular and neurobiological processes that regulate social interactions. This knowledge could help us better understand where to look when things start to break down. Social interactions are tightly linked to individual success. And in evolutionary terms, success is defined as an organism's ability to survive and reproduce. So that means that natural selection has shaped these traits. And it doesn't matter if we're thinking about humans or primates or even bees. Throughout the course of their natural histories, evolution has fine-tuned aspects of their brains to help them better navigate their social environments and interact with each other. And this set of changes is what scientists often refer to as the social brain. I'm an evolutionary biologist, and the goal of my lab is to understand the mechanisms that shape the evolution and the development of the social brain. Armed with this knowledge, we could learn general principles that give us new ideas of where to look as we develop new diagnostic tools or treatments for our own human social disorders. But how do scientists begin to understand the mechanisms that shape the social brain? Most of the time, we start by making comparisons among things that are the same. And then we conduct controlled experiments where we manipulate a single variable of interest while we keep everything else constant. And this allows us to isolate the effect of that particular variable. So you may be thinking about lab studies that we can do using fruit flies or mice. In these systems, you can take a group of highly genetically similar individuals, and you can give half of them a drug, while you give the other half a placebo or a control. And this allows you to have the experimental precision that you need to understand the effects of that drug on your focal population. But as a result, it also limits the amount of variation that you can study. And this is a problem because we can only learn so much from studying things that are all the same. In the case of social behavior, a narrow behavioral range limits the scope of the questions that we can ask. Mice, for example, exhibit some forms of social behavior like mating and maternal care, but they all share a similar behavioral repertoire. So what if we want to understand what generates the potential for social interactions to arise in the first place? Then we would want to be able to compare and contrast individuals that live in social groups with those that do not. Unfortunately, most of the model systems that we currently use are limited in this respect. While we've learned a massive amount from studying these systems, they don't adequately capture this range of behavioral variation. And that makes it hard to identify that set of essential ingredients that are needed for social behaviors to arise in the first place. So what if there were another way? What if instead of comparing things that were the same, we could start by contrasting things that are different? Let's say that you want to learn how to make a truly excellent cookie. Which is going to be more useful for you? 20 nearly identical chocolate chip cookie recipes that use the same ingredient set and vary only in the proportions of the ingredient that they're using? Or 20 wildly different recipes that use different types of sugars and different flavors and different add-ins? Miso chocolate chip, anyone? By comparing and contrasting these wildly different cookie types, you can start to identify new groups of what we call same differences. And these same differences can teach us what makes a good cookie. For example, if you were to compare and contrast dark chocolate walnut with white chocolate macadamia nut, you might be forced to conclude that sweet and crunchy makes an excellent dessert choice. And by casting a wider net, you can learn what it is that makes a good cookie in general. We can apply the same approach to biology as well. Let's say you want to understand how animals adapt to extreme environments. Maybe you would start by comparing animals that live in the Arctic to those that live in the desert. You can then identify traits that are shared among the Arctic living animals and contrast those with traits that you find in the desert living ones. Arctic environments are extremely cold, with lots of ice and limited vegetation. And examples of animals that live here include polar bears, Arctic foxes, and seals. In contrast, desert environments are extremely hot, with limited water and very sparse vegetation. And animals that live here include camels and fennec foxes and kangaroo rats. When you compare and contrast these different groups, you might notice that the Arctic mammals have a dense layer of fur and a thick layer of fat that surrounds their entire bodies. This allows them the insulation that they need to stay warm in the freezing cold. In contrast, the desert mammals have a thin layer of fur, and they actually concentrate their fat in specific body parts, and like camels that store fat in their humps. And this allows them to avoid overheating in that extreme heat. Desert animals like the fennec fox have these huge ears that they can use to dissipate heat, while the Arctic fox has really small ears to help them retain it. And desert animals are active mostly at nighttime to help them avoid the scorching sun, while Arctic animals can be active in both daytime and nighttime. So we can learn quite a lot by comparing groups of animals with similar traits and contrasting them with those that are different. We can use the same approach to understand social behavior better too, by comparing groups of animals that live in social groups to those that do not. And through this process, we can start to identify sets of traits that are shared among the social organisms and not found in the social ones. And this set of same differences can help bring into focus what it is that makes those social organisms unique or different. And this helps us identify that set of features that have been refined by natural selection over time to shape the evolution of the social brain. I use this approach to study a group of wild bees known as sweat bees. Unlike many of the commonly studied model systems, sweat bees exhibit a wide behavioral range that includes both social and solitary individuals. Some sweat bees live in social groups where you have a reproductive queen and non-reproductive workers, and others live and reproduce independently. Throughout their evolutionary history, sweat bees have gained and lost social behavior a number of different times, and this generates a lot of natural variation that we can use as a lens to compare and contrast their genes and their brains and their behaviors. When we compare social sweat bees to sweat bees that are solitary, we've identified a core set of genes that have been shaped by natural selection as social behavior was gained and lost. And some of these genes are thought to play an important role in hormone binding and transport of that hormone into the insect brain. We've discovered that social sweat bees have higher levels of this hormone in their brains compared to solitary sweat bees. We've also zoomed out and we've looked in ants, which is another lineage of social insects that diverged from bees over 180 million years ago. Again, in the ants, we find that variation in the hormone levels in their brain is correlated with natural variation in their social behaviors. And we also know that hormones can regulate social behaviors in vertebrates as well. I mean, we've all met a teenager. These observations suggest that there may be overarching commonalities in the mechanisms that regulate social behaviors across vast evolutionary distances. And this suggests that from bees, we could learn general principles that could apply to our own human social behaviors as well. Evolution teaches us that many of the genes that regulate the insect brain perform similar functions in the vertebrate brain. We actually share 80% of our genes with cows, and nearly 50% of our genes with fruit flies and with bees. Through our own work, we've discovered that at least some of the same mechanisms that regulate social variation in bees are also associated with variation in human social behavior. For example, when we compare genes that vary between social and solitary sweat bees of the same species, we find a significant overlap with genes that are linked to autism in humans. These results highlight how conserved mechanisms can shape variation in social behavior across vast evolutionary distances that span from insects all the way to mammals. And they give us an important thread that we can start to pull on to begin to unravel what those mechanisms are that make the social organisms unique. And they help us to identify that set of features that have shaped the evolution of the social brain. Of course, we know that our behaviors are more than a product of just the genes that we carry, but it's also the environments that we experience. And it's really that interaction between genes and the environment that generates individual variation in behavior. We know that the environments that we experience as children can strongly influence the way that we behave as adults. And we know that different parenting styles can have really strong influences on an individual's social skills in adulthood. So, if we really want to understand what makes and breaks the social brain, we need to understand not only the genes, but how these genes interact with the environment and how these interactions get encoded into the brain. And since we've already learned that we can understand social behaviors better by studying bees, why not compare and contrast how social and non-social individuals differ in their responses to social experience? We're already starting to do these experiments now to see if social individuals are more sensitive to their social environments compared to their solitary counterparts. And we're also interested in identifying which regions of the brain are the most sensitive to these differences in social experience. So far, we've conducted these studies in bumblebees, which all live in social groups. And when we isolate bumblebees from the colony, it changes the way that they interact with other bees. Insects use their antennae as their sensory windows to the world. And we think that antennal contact is an important way that bumblebees exchange information with each other. We've discovered that bumblebees typically interact through something that looks like an antennal handshake. They approach each other face to face, and then they briefly contact their antennae together. But when we isolate bees during a sensitive period in their early lives, they become much less specific in the way that they interact with other bees. Instead of these short, direct face-to-face interactions, they actually become much less predictable. It's as if you would meet someone the first time and you would go to shake their hand, and then the next time you try to shake their face. We've also discovered that isolated individuals have much higher levels of variation in how their brains develop compared to bees that are reared in social groups. And we've identified a set of genes that vary in expression levels depending on social experience. And some of these genes are the same genes that we identified earlier that are linked to hormone binding and transport into the brain. Social experiences strongly impact behaviors in a wide range of animals. And isolation can increase aggression and decrease sociability in species as wide-ranging as spiders and mice and primates. And in humans, we know that early life social deprivation is linked to a higher risk of neurodevelopmental and mental disorders in adulthood. So, by studying the effects of social experience, we can gain a complementary window that helps us understand how the social brain develops. To do these experiments, we've had to build the resources from the ground up. You can't order sweat bees on the internet, and there are no breeding facilities, so we have to go outside and we have to find them and study them in the natural world. And this has been challenging work, but it's been totally worth it because we're gleaning exciting new insights into the pathways that are associated with social variation in the system. Our next steps are to understand how these pathways function and to understand how they respond to different environmental inputs. And of course, we want to compare and contrast how social and solitary individuals respond to these differences. Through this process, we can start to identify that core set of mechanisms that make social organisms unique, and this can give us exciting insights into the underlying blueprint of the social brain. Social interactions are crucial to the lives of so many species. And when these interactions break down, the effects can be extremely costly. By studying animals that already vary in their social behaviors, we can learn exciting new things about that set of same differences that make social organisms unique. And this can give us new insights into the fundamental blueprint of the social brain that can apply to many species, from insects, potentially all the way to people. We can learn so much by studying the variation that's already out there in the world. as long as we're open to it. Thank you.